Morning, good afternoon, good evening, dear colleagues. On behalf of the group of Jan Dirk Grunwald, I would like to show you some results which the group has been working on during the last 7 to 8 years. And I'm very happy to present at the Global XAS Journal Club today on behalf of my colleagues. I will be talking about photon in, photon out spectroscopy techniques and XAS used on catalysis, heterogeneous catalysis, specifically now on automobile catalysts. And I will be explaining how we used these techniques to answer a few interesting questions on the ammonia SCR reaction, which I will get to in a few minutes. First, I will give, in the first part of the talk, I will give an overview of exhaust gas after treatment catalysis and specifically the selective catalytic reaction, selective catalytic reduction of nitrogen oxides, relate that to the use of synchrotron radiation, how it's used in catalysis, specifically for Operando spectroscopy, and introduce the method of Operando experimentation that we use. In the second part of the talk, I will give insight into what chemical information we can get from the spectral features of X-ray absorption spectroscopy and X-ray emission spectroscopy. And finally, in the third part of the talk, I will use the obtained knowledge, which I present in the second part, to further explore the so-called seagull profile, which in this selective catalytic reduction reaction is the dual maxima at two different temperature regimes, showing us that we have a dual reaction mechanism. So let's get started with the first part. In the modern world, we are faced by a large number of vehicles on the roads, and this is also causing a lot of environmental pollution. And in order to abate this pollution, both the automobile technology such as car, motor development has to be improved to emit less pollutants, and also the catalysts have to be improved to reduce or to decrease the amount of emissions. So in Europe at the moment is the 6C emission standard, which demands that all the pollution components are decreased. In this talk, I will focus on the abatement of nitrogen oxides. And in diesel cars, there are several catalysts installed in a row. So depending on the application, whether it's stationary or marine, heavy duty or light duty, we typically use different catalysts to reduce NOx with ammonia. So this is a selective catalytic reduction. So we have to selectively let the nitrogen oxides react with ammonia to nitrogen and water, which are completely harmless. We need the selectivity from the catalysts, and the catalysts which are used today are vanadium-based, iron-based, and copper-based on zeolites. How does this work? We have our exhaust pipe and the catalyst can, in which there's a monolith covered with a wash coat, and that is our catalyst, which is the zeolite. Here's one of the cells of the zeolite with the copper active sites, or the active sites, for example, iron. Now, the principle of diesel engine is based on the fact that there's that there is an excess of air. So we are always operating under air, under oxygen-rich conditions. What do we do at the synchrotron to study these reactions? So first, I should mention that the catalysis research at the synchrotron is as old as research at the synchrotron. So as soon as X-ray beamlines were available, already in situ experiments have been conducted through the 70s and 80s, and starting from the 90s, more and more complex experiments combining multiple techniques, synchrotron-based and non-synchrotron-based, with XAS, for example, have been implemented, as I will show later. So on a time scale, we have, starting from the first generation sources operating in parasitic mode to second generation reconstructed synchrotrons, further going to third generation dedicated synchrotrons, and further fourth generation sources, which we have today. We have now many synchrotrons in the world, and here are some European synchrotrons that are used by our group to do these studies. Now, to develop catalysts, we need some interdisciplinary efforts between the material preparation, the catalytic performance measurements, and once we want to find out what do we have in the catalyst that makes it active, we ask ourselves, okay, what is the structure? So we need to characterize the catalyst, and then to make sure that everything makes sense from a theoretical point of view, we add theoretical calculations, DFT, and then at some point we might have an answer to our overall questions. Why is the catalyst working, or is it not working? How does the structure relate to activity? And can we maybe improve our catalyst formulation? So based on this rational design cycle, I will give from each of the points some examples. So we'll be talking a little bit about vanadium, iron, and focusing more deeply on the copper-based catalysts. I'll be showing some catalytic testing, both in the lab reactor and at the synchrotron. Characterization by XAS, by photon in, photon out, Herft Zanes, and valence-to-core X-ray emission spectroscopy, and then justifying the results by theoretical calculations. So a catalyst, by definition, is a material which increases the catalytic rate without participating in the net reaction, but it is not a dead material. It is a material which actively and dynamically participates in the reaction. So we need to characterize it operando, that means under reaction conditions. And one of the mostly used operando methods is infrared spectroscopy, showing the surface adsorbates. Also for SCR catalysts, electron paramagnetic resonance spectroscopy is used, which is very powerful and sensitive to the active site and can characterize the local geometry and interactions in the catalyst, but is limited to only specific oxidation states. Of course, we have our favorite technique, X-ray absorption spectroscopy, which probes the element, specifically the whole catalyst. And here I should mention also towards the preparation of the catalyst, since Zanes, or since X-ray absorption spectroscopy probes the bulk, and we want to be surface sensitive because catalysis is a surface set of reactions. The catalysts here for this work were designed in such a way that we have isolated sites. So all of the sites, all of the copper or iron or vanadium sites, they are exposed to the reactant. Finally, we have DFT, which it can computationally put the full picture of the catalyst, so the active site, the zeolite framework, and the reactants into perspective. Now, based on these operando techniques, many authors have proposed various mechanisms which are very similar. This is NO and ammonia in, nitrogen, water out. There are some differences, of course, but we need to really be specific to what we're probing. So infrared spectroscopy is probing the surface species, but it might be unspecific to the adsorption site where these surface species are. EPR is and XANES are giving information on the catalytic site itself. But if we want to link these two, here I will introduce the valence-to-core X-ray emission spectroscopy, which is able to probe both the elementally specifically site, the catalytic site, and also the ligand, which is adsorbed onto it. And of course, this can be theoretically modeled by DFT. Now, the experiment itself, we have our catalyst in a plug-for-flow reactor, mimicking in some way the exhaust after-treatment system. This is a 1 millimeter capillary tube with a gas flow, controlled gas atmosphere. We have heating by the heat gun or resistive coils, and we shine the X-rays through the catalyst in order to probe it. Now, the experiment which I will show you here is based on a RIX setup. So here we have both the monochromator to monochromatize our incident X-ray beam, and also arrays of analyzer crystals to monochromatize the emitted photons, so the emitted fluorescence, so that we can obtain high energy resolution fluorescence detected XANES spectra, and also the high resolution fluorescence. The experiment is here the reactor with a controlled gas atmosphere, with mass flow controllers for the gases and analytics to find out, to analyze the gas composition at the outlet. A typical experiment is here. We have photons in, photons out. Since the selective catalytic reduction is quite a complex reaction, we want to study the individual components. So we decompose this into a set of gas mixtures. So oxygen containing only, some inert mixtures, some reducing mixtures, and the other ones ammonia containing mixtures, and the realistic SCR. I would like to pinpoint here that the nitrogen oxide and ammonia are on the 100 to 1000 ppm level, whereas the oxygen, water on the percent level. So we're always, in most cases, we're in net oxidizing conditions. Now, when we measure XAS, the information that we get is twofold. We have the XANES and the XAS, so the electronic structure and information on the neighboring atoms. That is basically two complementary pieces of information in a single scan. Now, if we go to the X-ray emission spectroscopy, we get an interesting picture. So the X-ray absorption measured by the fluorescence channel is basically the probing of unoccupied valence states here, giving us information about the coordination, local coordination, oxidation state, and local geometry. And if we do the emission, and that's not the strong emission here, the core-to-core, but the valence-to-core emission, which is a very weak transition, we're probing the occupied valence states as they decay to the core hole. And here we get the electronic structure of the low energy side of the Fermi level, and can also distinguish between the ligand type, nitrogen versus oxygen. And this is not possible by XAS, because XAS does not distinguish between neighboring elements. Some examples of the elements which I will be talking about here, and how what features we see in the X-ray absorption spectroscopy. So for example, starting from vanadium, which is a D0 or D1 system, we have many unoccupied states giving rise to this very high pre-edge peak. And then we're also sensitive to oxidation states. For iron, we can also distinguish between oxidation states and other transitions based on the pre-edge peak. For copper, it becomes a little bit more interesting because copper 2 is already a D9 system, so it has a very tiny pre-edge peak labeled as A, and copper 1 oxide is it doesn't have any pre-edge peak anymore because its D band is completely filled. But we have other features which are of interest. Now, before I go into detail with these, with the rest of my talk, I would like to take questions.
I had a minor technical question while we wait for people to type questions into the chat. If you could go back to slide 13. I was curious how confident you are about the consistency of the energy scale between the HRFD and the valence-to-core.
They align pretty well. That's a good technical question. Both the monochromators, of course, are calibrated, and also the analyzer crystals are calibrated very carefully to the energy. So when you put this spectrum with this spectrum together, they align very well.
It's the reason I ask is if it was a XANES rather than a HRFD spectrum, I would have expected that your large pre-edge feature B would align with a dip in the in that high energy tail of the valence-to-core emission. And so it looks like it's about half a volt off from that, right? The emission coming out of the sample gets partially absorbed by the XANES, by the mu of E. And so I would usually expect little dips and all the wiggles in the high energy tail of the valence-to-core to be inversely related to the absorption coefficient. Now, yeah, so it could well be that that's the case, but we can't see it here because HRFD, depending on where you are on your, I'm assuming a K alpha 1, it could shift things a little bit. But anyway, that's my question.
Okay, I have not really thought about how the two relate to each other in terms of exactly that they're complementary. That one has a maximum while the other one has a minimum. But okay. I would check that by calculations to see.
Sure. Again, it has to do with finite sample thickness.
Okay.
Okay. Laura has a point that maybe core hole potential can shift the spectra. I don't know. It would be an interesting thing to talk with the theorists about.
Definitely.
Seeing no other questions, you should continue, please. Oh, wait a minute. Matthew, do you have a question?
Actually, just a suggestion for you, saying that on your point, I was thinking that perhaps the reason you don't see that dip at the B position is that maybe the sample is dilute enough so that the probe depth doesn't change much at B versus A, for instance.
Yeah, but Matthew, there's no physics in the high energy tail of the valence-to-core. That should just be a Lorentzian continuation because of lifetimes. Is my understanding, though it could be that there's a breakdown in a single particle picture, and maybe we should be saying there's single particle occupied states above the Fermi level. I don't know. All right, Alexey, you should continue.
Stuff doesn't really look like the XANES.
Okay. So in my second part of the talk, I will go more deeply into the information obtained in the X-ray absorption, X-ray emission spectroscopies, and how this can be related to the chemical information in the system. So first, going directly to the vanadium, we have here our X-ray absorption spectrum and X-ray emission, we can see that these spectra are on each their side of the Fermi level. Both spectra here are showing different oxides with different oxidation states of vanadium, so vanadium 3, vanadium 4, vanadium 5. And we see that both spectra are very sensitive to the oxidation state. Now, under reaction conditions, taking just Herfti-Zanes, once we go to ammonia, which is a reducing environment, we see here the reduction of vanadium 5 to vanadium 4 from the edge position and also from the pre-edge. This can be further analyzed by a so-called variogram of the pre-edge region, where if the position of the pre-edge is plotted against its intensity, and we extract these data points from pre-edge features of different reference compounds and also of the catalyst, then we can directly estimate what oxidation state and what coordination number the vanadium in our system is. So, for example, here, the red points relating to the vanadium and the catalyst are somewhere between 5-fold coordinated vanadium 5 and 5-fold coordinated vanadium 4. And such an analysis is quite simple and gives a lot of information about the local geometry. Now, further information which can be obtained from the valence-to-core, other than the oxidation state, which we have seen. In the oxidation state, as we go to lower oxidation states, the K beta double prime peak, which is the satellite, it shifts and decreases in intensity. If we also, as I mentioned, that this method is sensitive to the ligand speciation, if we exchange ligands, so instead of an OH, we put an ammonia, so instead of oxygen, there's a nitrogen, the K beta double prime satellite shifts to higher energies. And by putting, for example, a carbon methyl group, it shifts further up. And to note that this feature is because vanadium is bonded to oxygen. So this is due to the oxygen bonding. Now, if we vary the ligands bonded via an oxygen, the variation is not so large as when when the ligands are varied in direct contact with the metal center. And this is completely consistent with some of the classical work by Serena de Beer, where she has shown that exchanging ligands shifts this K beta double prime feature. So under reaction conditions of a vanadium catalyst, we have some oxidizing conditions and then this reducing conditions with ammonia, asking the question, what is happening? So we see a slight decrease of the shift, like we've seen there, corresponding to the decrease of oxidation state of the vanadium, but there is no shift in the peak, for example, to higher energies, showing us that we do not have direct ammonia bonding to the vanadium sites. The same picture, same set of pictures we can draw with, in the case of iron, we have the pre-edge peak, which can be very nicely analyzed by a variogram, the position and versus the intensity. Here we have on the left hand iron 2, on the right hand iron 3. The lower intensities are 6-fold coordinated iron, that's octahedral coordination. The higher intensities, that's tetrahedral 4-fold coordination. Something in between would be 5-fold coordination. So depending on what reaction conditions the catalyst is put under, we are going between reducing conditions, that's somewhere at iron +2 and 5-fold coordinated, to iron 3 4 to 5 fold coordinated. Now, if we look at the Operando valence-to-core XES, we have also the main K-beta 2.25 line and the K-beta double prime satellite. And here we observe something like a peak and a very vague shoulder. So by doing many DFT calculations, trying to assign some structure to the shoulder, we came to a hypothesis that there is a partially positively charged threefold coordinated oxygen species with a protonated, which corresponds to this feature. So in order to see these features better, we took the second derivative of each of these spectra. And the different spectra can be related to different species here in this overall mechanism. Now going to the case of copper, we have two main features which are of interest. However, we have many features here at the X-ray absorption edge. We have the very vague pre-edge feature marked with A, and that corresponds to copper 2. And then as we reduce copper to copper 1, the pre-edge feature disappears, but that goes to the benefit of a very strong rising edge feature labeled B here. So A and B in the following will be used to differentiate between copper 2 and copper 1 in the system. Now, putting a copper zeolite catalyst under reaction conditions, we can see also under oxygen containing conditions, we have more of the copper 2 species. And at ammonia containing conditions, we have a lot of copper 1 being formed, but also to some degree, this copper 2 still being present because we are under net oxidizing conditions. And for example, some vague changes can be seen here under the helium atmosphere. Some copper 1 is being formed, so we have to some degree auto-reduction under inert atmospheres. Now, looking a little bit closer at the Zane spectra, we see this very weak pre-edge feature, that this is sensitive and can quantify to some degree the amount of copper 2, which we have in most of these conditions. And if we are under ammonia-containing atmospheres, copper 2 is still present. As we can see, it does not disappear completely, and only when we turn off oxygen completely, as in condition 9, this pre-edge feature really disappears. Now, taking the valence-to-core X-ray emission spectra and looking specifically at the main peak, which shows, of course, difference in features depending whether the conditions are oxidizing or more mildly reducing, ammonia containing. We have also the satellite feature, which is more shifted to lower energies for the oxidizing conditions and more to higher energies for the ammonia containing conditions, telling us that we have a different ligand type. And this allows us to directly assign ligation of ammonia to the copper sites in this reaction mechanism. Looking a little bit more closely at the satellite peak under NO, we see maybe some small variations here due to the NO in the reaction, but when we go to ammonia containing conditions, we clearly see this shift. So we can really distinguish both elements specifically, that this is copper, and also which ligands are bonded to the copper site. Now, to summarize the second part, I would like to repeat again which spectroscopic features can be related to the chemical species in our catalyst. We have the pre-edge feature of vanadium, iron, and copper to tell us about the oxidation state, or if the pre-edge feature here is missing, it's the rising edge feature. Oxidation states can also be obtained from the valence-to-core to differentiate, is it a ligand effect or is it really an oxidation state effect? For vanadium and for iron, we have this intermediate with the positively charged hydroxylated oxygen. And for the copper case, we are distinguishing between oxygen and nitrogen. Now, before I go further to the final part of the talk, applying this to a specific case study, I would like to take questions again.
There are questions, and I'll encourage everyone, please type them into the chat. Yang Ha, you have a question?
Yeah, so your XAS analysis, I see your analysis on the pre-edge. So my question, I have a technical question. So how sensitive is this pre-edge intensity to your data normalization, especially for copper when you have a very weak pre-edge?
Excellent question. I think I will be showing some spectra later. So these spectra are in the HERV-D zeins. So these are really highly resolved. So even a very weak feature is seen quite well. I will be showing later some spectra which are measured using conventional zeins in transmission or fluorescence at the normal XAS beamline, where this pre-edge feature is almost not detectable. You can see it when you really analyze your spectra.
Okay, and also, what's the loading ratio of your metals on the zeolites?
Yes, for vanadium, it is for all of these three systems. They have been really kept so low to keep 100% dispersion of the metal. So for the vanadium, it was do not really have around the 1%, but to have a sub-monolayer coverage. For iron and copper, it's 1%, and for copper, we also go below to 0.5%.
Okay, thank you.
I have a question or two about your valence-to-core emission, which is lovely data. How far above the binding energy did you put your incident beam? How non-resonant are you is kind of the question here.
Oh, it's been put well above the edge. So well, if the edge is at 9, then put it about 9.5.
Okay, so you definitely then could be exciting double electron features, which will show up on the high energy tail of the valence-to-core emission. Okay, all right,
well, that
very
is
good.
Possible. Okay,
yeah.
Maybe that resolves a few of the earlier questions.
Possibly. I mean, the way to check, of course, is to try different incident energies. You had this really interesting idea that there could be a connection between a change in oxidation state of the oxygen and what you're seeing in the vicinity of the beta double prime feature. I've forgotten. Yeah, around here. I haven't seen that discussed before, and I think it could be really interesting, maybe especially in the battery community, where the possible role of oxygen in the redox process itself, of course, is a hot and interesting and debated topic. Have you seen discussion of this issue before with respect to valence-to-core XES?
No, this was quite a struggle to really assign this feature, because it definitely is seen. And only very few DFT structures have actually identified such a structure. So if we have this NH2 bonded to an oxygen, to an iron, if we just place a proton at the top of it, then this feature can be calculated. But otherwise, it's only a 2-feature spectrum.
Okay. Thank you. You should continue, please.
Okay, so here's the final part of the talk. Based on all the information that we have from X-ray absorption and emission, I will now focus on the copper side, on the copper-based catalyst, to explore this seagull-shaped profile of the conversion curve. So normally, we are interested in quite a narrow range of temperatures when we operate the catalyst. So, but if we scan over a larger temperature range and study, for example, different loadings of copper, which are both low, but I will refer them to the lower loading and the higher loading, we see that there is a very significant difference between the conversion at low temperatures, especially. So at high temperatures, we gain somewhere close to full conversion, but at low temperatures, there's something which strongly depends on the loading of the catalyst. And this was discovered by several groups, and we can here say that there are 2 mechanisms. So it's not just a simple light off, it's really a 2-mechanism reaction. And let's try to find out, based on the spectroscopy, why this is being observed. So first, by applying different ratios of the reactants, so ammonia and NO, which are usually fed about stoichiometrically, we see that for lower temperatures, the reaction rate goes down as we increase the ammonia content. And that's both for the lower loaded sample and for the higher loaded. So at high temperature, we have quite high conversions. Now the question is, we see that at lower temperatures, ammonia is a poison because it decreases the rate of reaction. Now we want to find out which species, which sites in the catalyst are actually being poisoned. So in order to do that, we run the SCR reaction at the synchrotron and measure the Zane's spectra, ramping on the temperature scale, which refers to this seagull profile, and we see a number of changes. Now here, we cannot clearly identify clear isosbestic points, which tells us there might be more than 2 species. So it's not a transition from A to B, but maybe A to B to C. We suspect that during the ramping of temperature under SCR conditions, we are going somewhere from copper 2 to copper 1 with adsorbed ammonia and copper 1 without adsorbed ammonia. And to find out, what we do is apply multivariate curve resolution analysis to separate our spectral series into independent components, into independent spectra, which can then be recombined by linear combinations to our original spectra. And indeed, we see that there is this red curve, copper 2, which can also be experimentally measured under oxidizing conditions. Then we have the copper with ammonia, which is also measured under corresponding conditions. And then we have a third component, which is copper 1 without ammonia, which cannot be measured directly as pure spectrum, but which is then obtained as an internal reference from this multivariate curve resolution as a third component. So these analytic techniques are very valuable to obtain internal reference spectra, which can otherwise not be measured experimentally in pure state, and especially not purchased as a well-defined sample. Now of course, I have to mention that all of these species have been justified by theoretical calculations by DFT, so they are really reasonable. Now, doing linear combination analysis of the different spectra, here we start with the conversion curves, low conversion going to higher conversion for the lower loaded copper, and the seagull dual mode effect with the higher conversion. Now, as we ramp up in temperature, we reduce copper 2 to copper 1. So the copper 1 percentage increases. Also, we have an increase first of the copper 1 with ammonia, and then a decrease as we go to higher temperature. Ammonia desorbs, and the rest of the components is copper 2, which then decreases, which I have not shown here for clarity. What is now puzzling is that the poisoning by ammonia takes place in very similar manners on both catalysts, and the reduction of copper from 2 to 1 is also very similar in both cases, but the activity is very different. So then it must not depend only on the oxidation state or the ammonia poisoning, but more on the loading. Now, for the lower loaded catalysts, the monomeric copper sites, so the single monomers, are favored. Once we go to slightly higher conditions, also dimers can be favored. So the nuclearity of the copper sites, whether they're monomeric or dimeric, matters, and this can then be nicely studied by XAS. So at 200 degrees for the lower loaded copper and the higher loaded copper, under oxidizing conditions and under ammonia containing conditions, we have these monomers for the lower loaded catalyst, and we see only the copper oxygen shell with basically no further neighbors. Once we go to the higher loading and go to the oxidizing conditions, we see this second metal-metal shell, which can be analyzed as a copper-copper interaction, giving us the conclusion that we have dimers. But once we introduce ammonia, these dimers are split up into monomers and are being poisoned. So this is what we have at lower temperatures. Now, if we go to higher temperatures, we have quite an interesting picture. Under ammonia-based conditions, we still have monomers. We only see the oxygen shell. But as we go to oxidizing conditions, we see that we have dimers strongly interacting with the zeolite framework. And the same picture we have also for the high loaded catalyst. So we have restructuring of the catalyst, and we can really nicely differentiate between the position of the copper and its nuclearity in each of these cases by XAS. Now, putting it into the full perspective, we have now a table with lower temperature, higher temperature, ammonia containing conditions, oxidizing conditions, and then the realistic SCR conditions in the middle, and each of the cells of the table address the lower loaded and the higher loaded catalyst. Now, at lower temperatures under ammonia conditions, we have a splitting up of dimers, or we have only monomers which are being poisoned. Here, under oxidizing conditions, we have monomers which are not being poisoned, and we have these dimers being formed in the higher loaded copper zeolite. And under SCR conditions, we have this inactive ammonia poison monomer, but when we have dimers, they interact with ammonia, of course, because they have to run the reaction, but they're not anymore poisoned, and these show to be highly active for the reaction, for the higher loaded. Now, repeating the same with higher temperatures, we do not have ammonia absorption. We have monomers here, dimers here for ammonia containing and oxidizing conditions, respectively. And under ammonia SCR reaction conditions, we have both monomeric and dimeric sites, which are sufficiently active at these higher temperatures. Now, to conclude, to give an overall conclusion, we have seen here, I'll wrap up again, the seagull effect, that we have two temperature regimes, the lower temperature and the higher temperature regime. At the lower temperatures, we observe inactive monomers poisoned by ammonia, and the active dimers for the higher loaded catalyst, which are interacting with the ammonia and running the reaction and are not being poisoned. And for the high temperature regime, in both cases, we have these dimers. Also, a general conclusion about the importance of the Operando X-ray absorption and the X-ray emission spectroscopies. Why is it Operando? Is because we are measuring under real reaction conditions. We are obtaining the kinetics, which are taking place on the catalyst, and also, and only when we have seen that the ammonia is poisoning the catalyst, we can study this effect further and really correlate this poisoning with the nuclearity, with the activity. By valence-to-core emission, we have seen this very important observation for copper, that there is a direct ammonia interaction with the copper site, unlike the case of vanadium or iron, which is very important if we want to understand the specific case of copper. Now, Zanes have shown us that ammonia bonds to both catalysts. This is what I've shown with the multivariate curve resolution analysis. And the XAS is complementary, but because it differentiates between monomeric and dimeric copper sites. And finally, we can conclude that the higher loaded catalyst zeolites are supposed to be the more preferred for ammonia SCR. So putting all the puzzle pieces together, spectroscopic techniques with kinetics, we get the, we understand the importance of Operando spectroscopy. Now, of course, in this talk, I've presented results from several years of work by many colleagues. So I would like to thank all of the people who have, who I and my colleagues have been working with, supporting us at the beamlines, at the DFT calculations, providing, synthesizing the catalysts. I also acknowledge all the funding sources and the synchrotrons we have used for doing these measurements. And I thank you for your attention, and again, open for questions.
Thank you, Alexey. It was a terrific talk. We'll start with a question from Tim Hyde.
Hi there. Fantastic talk. Very ambitious to put vanadium, iron, and copper all in one talk, and very impressive. My question goes back to the vanadium SCR catalysts. Am I right in saying that there's no real correspondence to any specific vanadium oxide? Is not vanadium 3, 4, or 5, or the geometry is not 3, 4, or 5 from what you were showing there? Have you sort of measured any other model reference compounds to be able to hone in on what exactly the active vanadium species is in under reaction conditions?
So I have not been myself involved in the vanadium case.
Right.
So maybe some of my answers will not be sufficient.
Yeah,
but you
so
mean,
I
for
guess-
example, this.
For example, that
right?
one. Yes, that's right. Yeah.
The estimation of the oxidation state and the geometry. So which reference compounds are you mostly referring to?
It seems most of your reactive or under reaction conditions in red are sort of in the middle. They're between the 4+ and the 5+ references. It's a very nice way of describing that, but obviously, yeah, with Herf-Zanes in particular, it would be very nice to be able to hone in further as to what the geometry is, or what the true oxidation state is if it's some sort of mixed species. Or have you had to rely on Raman and other spectroscopic techniques to get those answers?
Okay, as I mentioned, I was not involved in this work specifically, but what I can state is that vanadium analysis based on just this variogram is very complex, because you have both the shift of the centroid position and the intensity at once. So a very accurate quantification just based on this variogram is not very reliable. It gives you an estimate. For iron, it's more easy to do, because this is a very well-defined rectangular variogram where you can, but it's also, it has its own limitations. So you can put these values in the variogram to get a first estimate, but of course you would need to model somehow or look further directly into the spectra themselves. So for example, in this case, here it was identified as vanadium 5 and vanadium 4. Vanadium 3 has also been shown. So this is a fingerprinting technique, the Zanes, and you have to compare with the reference compounds that you have. And the catalyst is not a bulk oxide.
Yeah.
Right. So there has to always be a lot of caution. And the comment that you made on using other techniques, yes, of course, the sample has to have seen other techniques before it goes to the synchrotron, because the synchrotron is able to answer some questions that other techniques cannot. But you need to know more about your catalyst in order to really model it or formulate what question you want to answer.
Sure. Okay. Thank you for your answer.
I'll add that your colleague Dmitry Dorenkin pointed out that for the vanadium, the oxidation state was quantified with K beta as an additional contributor. But moving on, Yang Ha, you had a question?
No, I don't. I just had a comment on the vanadium reference.
Okay. Peter and Jean, you have a question?
Yeah, it's related to the assignment of the poisoning by ammonia. So I was just wondering why. I was just wondering if copper nitride was identifying this, because based on my own experience, if you heat actually copper in the presence of ammonia, you may end up with some nitride species. So, but here you basically at this temperature only reported copper, that is nitrogen adsorbed on copper, and then copper plus. And so I was wondering if you also looked at possibility to have like copper nitride, really real bond formation between nitrogen and copper.
So there is interaction between nitrogen and copper, but this is nitride, you mean NCUN,
yes.
right? But
yes.
What about the other species? Because the copper is also surrounded by other ligands. Under which conditions would you would you form this nitride?
Because you supported the copper on zeolite, right?
Yes, it's on a zeolite. Yes, it's surrounded by an acidic environment. It's under net oxidizing conditions, and it is also under, let's say, moderate temperatures, so 200 degrees, 300 degrees.
His copper is very diluted. The loading ratio is around 1% or even less.
Correct. But yes, I would like to, I'm curious, what do you need to form this copper nitride?
No, I'm just asking in the fingerprinting and testing whether you also consider that, the spectra that you try to fit, whether also like, just considering copper nitride. I, as a material chemist, we first of all think about this because I do know that copper and ammonia can have this kind of strong interaction at that such temperature to form nitride. So, and there are actually some reference copper nitride compound that you can buy to test. So I was just asking if you tested, if you checked that that's not what you're forming.
Okay, to form a nitride, I cannot really say if it's being formed and is reversible, it can be removed as easily when you remove the ammonia flow. Of course, it cannot be excluded, but this is not a model which we have considered.
I'll just add to that when we look at valence-to-core emission in solution, and we're looking, for example, at ion pairing where there's not really a chemical bond, the beta double prime feature is really quite weak. And of course, we've only done that on a few systems, and maybe other people have observed a substantial beta double prime. So it's a fair question that if you're seeing a nitrogen beta double prime feature, does that require a strong overlap of electronic wave functions, such as with bonding, as opposed to physiabsorption? So I think it's an interesting question, but I certainly don't understand the catalysis chemistry. That was just a spectroscopy comment.
Okay, yes, so overlap. I mean, if you have coordination of ammonia by lone pairs of the nitrogen, is that the same as?
I don't know.
As having a nitride?
It'd be fun to have someone run some NW Chem calculations on those different small cluster coordinations and see if they had a feeling for how strong the nitrogen beta double prime would be between a nitride as opposed to having the ammonia there.
Yes, I would guess you would have the overall Zane's shape completely different because nitrides are not the same as copper in an oxidizing environment.
Yeah, I think that's also why I was asking the question because if you just had like an adsorbed ammonia on copper, then I was thinking, okay, well, can it lead to such kind of spectral shifts, or is it really strong interaction between ammonia, which in that case can also happen with copper, or really a nitrogen bonded copper? I've seen this before, so that's what I'm asking about. But yeah, so
okay. I do not know these systems, but what I would expect is, of course, you would have the shift because of the interaction, but also the rest of the spectrum should change dramatically. And the question is this copper nitride then as easily decomposed as when you remove ammonia, for example? Because this is a very rapidly and very reversible process.
Okay, thank you.
I don't know if that's the case if you have a nitride formation, if that would not be more stable. We just